Hello, this is Ashim Dhakal, and I'm going to talk about nanophotonic and photonic technologies being developed in Nepal. I thank the organizers for this opportunity. In this talk, I will discuss three things. First, I will try to explain why we need to focus on the cutting edge or advanced research, contrary to the common assumption that we should not because we cannot afford to do that. Second, I will discuss a new type of nanophotonics device called on-chip axicon-like lens, which we are developing in Nepal. Third, I will discuss a systems level implementation of fluorescence spectroscopy to detect harmful contaminants in drinking water. I will not discuss challenges and opportunities as I expect this has been or will be discussed adequately in other sessions. Let me start with so-called Global Innovation Index. So this index is based on several indicators to measure the health of the research, development, and innovation ecosystem in different countries. According to this index, not surprisingly, Nepal is in the red zone in the globe, indicating its poor innovation ecosystem compared to the rest of the world. After being ranked 108th, 109th in the previous years, Nepal has jumped to 95th this year. So this was touted as a major achievement by several people involved in this area. However, the reality is quite different as seen in this graph, where all indicators except for the business, market, and investment environment are quite poor. Our rank seems to be clearly biased by the business environment rather than the innovation outputs. So nevertheless, all of this indicates potential investment opportunity should we innovate, which is good for, which may be good for innovation. As clearly indicated by our very poor rank in the knowledge impact metric, as shown in this table, there is no room for satisfaction for the scientific community. Also, it is important to note that R&D output indicators seem to be poorer compared to the R&D input. So this does not bode well for the long-term development of the innovation sector, as it discourages investment, even though we have a good investment indicators. So therefore, clearly, we need to work on knowledge impact, which can only be enhanced by research at the cutting edge. So this is the main reason why we at the Phutung Research Institute focus on research at the cutting edge, in spite of the common pessimism in this pessimism concerning advanced research. At Phutung Research Institute, we mainly focus on advanced nanophotonics and optical technologies. We have Nepal's only nanophotonics and photonics lab. The lab is world-class in the sense that we have state-of-the-art infrastructure for design, simulation, and measurement. So here, so these are the chips we sometimes brag for being Nepal's first sets of advanced nanophotonic chips. I will discuss about one particular component of that is of this chip. So in the slide, you can also see the chips when compared with a 1 rupee coin. So you may ask, what exactly is nanophotonics integration, and what is the technology we have been developing? I assume that you are aware of the electronics integration, integrated electronics. Basically, all of our mobile phones, computers, they have these electronic chips. So the photonic chip is similar. So in an electronic chip, electrons or current runs through the structures in the chip. The idea is similar in nanophotonic integration or photonic chips, where, however, light runs through the structures instead of the current. Using the integrated photonics technology, we are trying to miniaturize an optical coherence tomography system, which is currently a benchtop system. So we are trying to miniaturize the OCT system into a very small size of size 1 by 3 millimeters. So the benefits of this integration, like in the case of electronics, is that it it will have a smaller size, it will hopefully be more efficient, low cost, and it will be portable so that the system can be taken to the point of care, for example, in remote villages of Nepal. So now I will discuss a key component of the on-chip OCT that was there in the chip I showed before. So in a traditional free space setup, so-called axicons could be used. The axicon has a conical surface on the one side and the planar surface on the other side. So when the light beam impinges from the planar side, the light refracts to the axis. So all the light reflects to the axis, creating a focus of long length on the axis due to interference effects. So the resulting long depth of focus allows for scanning deeper tissue structures. So in the case of a photonic chip, where the light is largely confined within the chip, however, this traditional approach based on free space light cannot be used. Cannot be used, and also the delivery of light to the tissue and collecting the light back from the tissue to the chip. So delivery of light from the chip to the tissue and back from tissue to the chip is a very challenging problem in integrated photonics. So to address this problem simultaneously, we have developed an on-chip or chip-based axicon lens, which works on the principle of diffraction of light, like in the gratings. So here we saw our design, the microscope image of an actual lens and the expected behavior on top of the photonics chip. So the details, I'm not going to discuss quite a lot of details about this particular work because it's available in archive. So if you are interested, you can look at this. And this work is currently, the manuscript is under review. Here is the measured behavior of our chip, our axicon, which we measured in Nepal, of course. So we have a central lobe of 5.7 micrometer diameter, which spans over about 1 millimeter. So which should be sufficient to penetrate 1 millimeter depth of the tissue, hopefully. So in this slide, I'm sort of trying to give a glimpse of how the design and measurement are interlinked. I hope you have appreciated that we have developed a nanophotonic design and a measurement ecosystem in Nepal. But I have to tell you that we do not have the facility to fabricate the chip itself. So this is largely done by our partner in the UK by Cornerstone Project. The fabrication of the chip is a very expensive enterprise, and it needs a long-term big investment from the government, most likely. But rest of the things we can do it in Nepal. We have been doing this in Nepal. All right, so now I move on to discussing another project where we have been developing a fluorometer to detect contaminants in drinking water. So when exposed with a UV light, almost all the minerals and biochemicals exhibit fluorescence in different colors depending on their molecular composition, as you can see in the figure there in the left. So the key idea of this project is that the specific impurities in water can be detected by measurement of the light of the fluorescent light at some specific fluorescent band. So this is a schematic of our basic setup. So what we essentially do is we focus the light from the UV LED into the water sample, and the fluorescence is then collected and then detected by a photomultiplier tube or a spectrometer using a series of lenses, optical filters, gratings, and mirrors. So this is schematic just to give you a flavor of the optical design, not exactly what we have done. It's a bit complicated than what I've shown here. So this slide shows the measured calibration curve of our setup. the calibration is done using tryptophan amino acid, which is indicative of the biological activity in the water sample. So in a natural state, if we detect some tryptophan, it indicates that there is some bacteria, there is some biological activity going on there. That's why we choose tryptophan. So as you can see, our setup can measure as low as 100 parts per trillion in water. So this detection limit is better than most of the commercial fluorometers available in the world. So tryptophan, so these are the fluorescence spectra as measured with our system. As you can see, we can measure the spectra of tryptophan as low as 1.25 parts per billion in water. So here we have attempted to to measure these fluorescence from the actual groundwater samples taken from different locations in Kathmandu Valley. The yellow bar labeled as drinking water, which exhibits the lowest fluorescence, is from our reverse osmosis filtration system. So water from the groundwater from several places seem to exhibit fluorescence corresponding to tryptophan of concentration 7 to 28 ppb, while that of the RO system is lower than 1 ppb. So we are currently working on correlating these findings with microbial studies. So this is an ongoing work, and hopefully in the next conference I can discuss more about it. So with this, I would like to end by acknowledging our research partners, our funding agencies, and our sponsors, so without whom, of course, this whole research would not have been possible. So with this, I thank you, the audience, also for listening, and I'll be very happy to answer any questions you may have. So thank you very much.